DNA structure. Let's talk about the composition of nucleotides. This is a phosphate. Attached to our phosphate, there is a deoxyribose. And attached to that deoxyribose is a base. So this is the composition of a nucleotide. Remember our base? Well, there are 4 kinds of bases: adenine, thymine, cytosine, and guanine. Here is our nucleotide. As you can see, the base is linked to the deoxyribose and the deoxyribose is linked to the phosphate. But with what? They are all linked to each other with covalent bonds. Here are our bases again. They do something called complementary base pairing. Look at adenine and thymine. They match. And so do guanine and cytosine. This is called complementary base pairing, as each base can pair up with only one specific other base. The bonds linking bases together are called hydrogen bonds. Linking bases together links nucleotides together, which eventually forms 2 attached DNA strands. Then these 2 strands wind to form a DNA double helix, which looks just like the picture. DNA replication. What is DNA replication? Well, it is when an old DNA double helix breaks down to form a new DNA double helix. For the first part of this process, a DNA double strand and a helicase molecule are needed. The double helix unwinds and the helicase molecule proceeds to separate the 2 strands. The second part of this process is the making of new DNA double helices. For this, all DNA single strands, DNA polymerase are needed. DNA polymerase brings new strands that are matching the old strands in terms of bases. Once the matching strands have found each other, the DNA polymerase molecule proceeds to wind them with each other. Once this process is done, the new double helices are formed. Complementary base pairing, which we have already talked about, is very important. If one base is linked to the wrong base, such as adenine being linked to cyanine or guanine, instead of thymine, it codes for a malformation in the body, a genetic malformation. It is said that DNA replication is semi-conservative, as one old strand and one new strand are linked together to form a new double helix. Transcription and translation. Let's look at the differences between DNA and RNA. The first one is that DNA is composed of a double helix, while RNA is only composed of a single strand. Another one is that DNA contains a deoxyribose in its backbone, while RNA only contains a ribose. The last difference is that the base, thymine, the one matching adenine, does not exist in RNA. It is instead replaced by uracil. So these are the 3 differences you need to memorize. Let's now talk about transcription. For this process, you need to unwind the DNA double helix in order to be able to use a single strand of it. And you also need a molecule of RNA polymerase. Then, the RNA polymerase starts to form an RNA strand by using the DNA strand as a template. This is done through complementary base pairing. Here, you can see the process. Once the process is done, the double helix rewinds again. This new strand is called messenger RNA, also known as mRNA. 3 bases form a codon, and each codon codes for a particular amino acid. These amino acids that we are talking about form proteins for DNA and RNA. Translation is the process of protein synthesis. Ribosome, mRNA, and tRNA, also known as transfer RNA, are used during this process. tRNA is composed of anticodon, which, just like codons, is a triplet for RNA. First of all, mRNA binds to the ribosome. The amino acid attached to the tRNA corresponds to anticodons. tRNA also binds to the ribosome, but only the matching anticodon to the codon of the mRNA can do so. Complementary base pairing is used once again. Although, don't forget that in RNA, thymine is uracil. Just like two matching bases of DNA, two matching bases of RNA will be linked with a hydrogen bond. Once the whole codon is bonded to the whole anticodon, the amino acid forms a peptide bond. This process goes on until all the codons of the mRNA are linked to their matching anticodons. This forms a polypeptide chain, and it is the basis of protein synthesis. Now, what is the relationship between one gene and one polypeptide? When a peptide bond is formed, a polypeptide is also formed. There are a wide range of possible polypeptides. Genes store the information required to make polypeptides. The information is formed in the codons. The sequence of bases in a gene codes for the sequence of amino acids in a polypeptide. Enzymes. The blue shape represented is an enzyme. But what is an enzyme? Enzymes are globular proteins which act as catalysts of chemical reactions. As you can see, something called an active site is pointed out in the picture. An active site is a region on the surface of an enzyme to which substrates bind and which catalyzes a chemical reaction involving the substrates. Each enzyme is specific to a certain substrate, so the active site of an enzyme only works with the specific substrate. Now, let's look at the effects of temperature on enzyme activity. As you can see on the graph, the activity increases with the temperature, but once the enzyme has reached its optimum temperature, it starts denaturating. This graph shows the effects of pH on enzyme activity. The optimum pH of an enzyme is the pH at which the enzyme works best. As the pH diverges from optimum, the enzyme activity decreases. Acids and alkali environments can denaturate enzymes. Finally, let's look at the graph representing substrate concentration on enzyme activity. Enzyme activity increases as an increase in substrate concentration. But once the point of saturation of the enzyme is reached, although the substrate concentration might still increase, the enzyme activity won't. Enzymes cannot denaturate due to substrate concentration. Denaturation is changing the structure of an enzyme so it can no longer carry out its function. In lactose-free milk, the enzyme lactase is used in order to break the lactose down into glucose, so lactose-intolerant people can still drink milk.
To conclude this PowerPoint presentation is the last but not least important subtopic, 3.7, which is cell respiration. 3.7.1 and 3.7.2 asks to define cellular respiration, which is controlled release of energy from organic compounds in cells to form ATP, and to state that in cellular respiration, glucose in the cytoplasm is broken down by glycolysis into pyruvates with a small yield of ATP. 3.7.3 and 3.7.4 explain the process of cellular respiration. During anaerobic cellular respiration, pyruvate can be converted into the cytoplasm into lactate or ethanol and carbon dioxide with no further yield of ATP. It's less effective in oxidation than aerobic respiration. Lactic acid is produced in humans and is a product, and ethanol and carbon dioxide are produced in anaerobic yeast respiration. Lastly is aerobic cellular respiration. The pyruvates can be broken down in the mitochondria into carbon dioxide and water with a large yield of ATP. The ATP production in aerobic respiration is much higher than that of anaerobic respiration. Oxygen plays a large role in this. The water that it creates is absorbed into the cells, and the carbon dioxide is expelled from the body. This last slide shows a helpful diagram of cellular respiration that can be used in your notes for further reference.
I hope you enjoyed this magical journey through Chemistry of Life.
Feel free to copy down any of the information from this presentation in your notes.
Thank you for listening.